true third-line patients, and some of the patients can have received additional lines of therapy as well. It's a randomized study, and it's randomized on a 2-to-1 ratio, so that most of the patients will actually get the active drug, half the other third of the patients get best supportive care alone, and then at the time of progression, they're unblinded, and if they've got placebo, then they can cross right over to the active drug. Let's see. So again, and this was just the entry criteria. So the primary endpoint of this study is progression-free survival, and of course they're going to look at overall survival and time to progression and so forth. And this is actually very similar to the study design that got sunitinib approved in the second line. So it's that's kind of their strategy. And for the biostats for this, basically what they're trying to show is an improvement in progression-free survival from 6 weeks with placebo to 12 weeks with active drug. They're planning on enrolling a total of 170 patients, and they think that this can be done within 12 months. So the study is just starting accrual, and hopefully we'll have completed accrual within a year or so. Again, this study is basically for this group of patients here. And what I've listed here is just the standard treatment options for you with imatinib first line, sunitinib second line. I list here sorafenib just because it's one that's clinically available. But this is the population that regorafenib is trying to be placed. So if it can get that 100% improvement in time to progression, this is going to be the indication they're going for. What you can also see here, as you know, is that although you can get really good response rates in first-line imatinib with a prolonged progression-free survival of about 2 years, the response rate as well as the progression-free survival for patients on second and third-line drugs are quite modest. And so the question is, how can we improve that? And that leads to kind of the second program that we're developing and working with Ron, and that's the study of dasatinib and ipilimumab. The question here is, again, can we harness the immune system, maybe enhance the immunologic effects of tyrosine kinase inhibition, or alternatively, can we enhance the effects of immunotherapy with a drug called ipilimumab by targeting and perhaps causing apoptosis of the tumor cells? Dasatinib, as you know, is another tyrosine kinase inhibitor. It was actually developed as a SARC-enabled dual kinase inhibitor, but also hits KIT and PDGFR alpha and a number of other targets. Ipilimumab is a drug that you may not have heard about, though. This is some of the immunological effects that I'm not going to go into because Ron did such a great job, but what's become clear is that the tyrosine kinase inhibitors do more than kind of just what we thought. So although they have effects against the tumor cells, they also have effects against the normal body cells, including the immune system. And some of these effects are really just recently being elucidated. We're working very closely with Jed Wolchuk, collecting blood and tumor for patients treated with these various drugs so that we can actually look at what's happening to the immune system for patients on Sutent or imatinib or any of these other TKIs. So ipilimumab is a drug, as Ron said, it targets something called CTLA-4, and this is a complicated immunology slide. But this is an important drug because it's really a first-in-class that targets these things that we call the immune checkpoint system. And it was recently approved just a couple weeks ago for melanoma. It's the first drug to ever show that it improves overall survival in melanoma. No drug has ever shown that before. If you look at the number of patients alive at 1 year and 2 years, it's almost double for patients who get ipilimumab versus those who don't. So this is a very important drug. It was tested first in melanoma because melanoma is what we classically consider to be an immunologically relevant disease. Is now just being recognized as such. But Gleevec or ipilimumab, this slide is complicated, but let me just walk you through it because this can maybe help understand what this drug does. This here on the top these are T cells. These are the good cells, basically, CD8 cells. And these cells on the lower row are something that we call antigen-presenting cells. In order to turn on the immune cell, the good cell, it has to be activated. And to activate it, it needs kind of two signals that occur between interactions between this antigen-presenting cell and the T cell. And the first occurs when some antigen that the immune system is supposed to recognize is presented by the antigen-presenting cell in this complex here, which is the T cell receptor and this MHC complex. And if we use an automobile analogy, once that connection occurs, this is like putting the key in the ignition of a car and get things started. But for full activation, you need a second signal. And that signal occurs when this thing called B7 on the antigen-presenting cell interacts with CD28 on the T cell. And when that happens, you get full T cell activation. The T cell can go on and attack tumor, do whatever it needs to do. And this is kind of like putting the foot on the gas pedal. Okay? But CTLA-4 is kind of important in suppressing that function. And it's important normally, otherwise you might get autoimmune diseases and so forth if the immune system's hyperactivated. So CTLA-4 is this protein here in the T cell. And once the T cell is activated, it actually moves from one side of the T cell to the other here. And once it's here, it can interact with the antigen-presenting cell, and it actually binds with this thing called B7, which is supposed to be binding with CD28. And it binds to B7 with greater affinity than does CD28. And once this binds, this inhibits the T cell. And this is like putting the brake on the immune system. Okay, so what ipilimumab is, it's an antibody that actually binds to CTLA-4, and it doesn't allow it to bind to B7 here. So CTLA-4 can't shut off the T cell, and you get persistent T cell potentiation. So that's what this drug does. You know, certainly this is it's kind of a first in class. There's another drug that blocks this by Pfizer, and there are kind of next generation drugs that target different parts of the immune checkpoint system. These are some pictures. We haven't used this in GIST, but these are some pictures from patients with melanoma showing kind of the types of responses that we see. And interestingly, because this is an immunotherapy, it works different than chemotherapy or imatinib or so forth. With imatinib, we're used to fairly rapid responses. But here, since we're affecting the immune system, it takes some time for it to work. So we have to give the drug has to alter the immune system, then the immune system has to attack the tumor. And what that means is sometimes we see tumor growth before we see regression. And you can kind of see this here. This is a patient with melanoma who has these flank nodules there on the skin, and you can see it on the CT scan there. And after 12 weeks of therapy, it's definitely worse. But without any further treatment, 4 weeks later, you can see it goes from that to that. And on the CT scan, you can see it's kind of coming outside there, and it's flattening out. This patient got further doses of therapy on 12-week intervals, and you can see by week 72, there's just a little bit of pigmentation there, but no viable tumor, and the CAT scan's completely normal. And I've got another picture. This is a patient here who, you can't see the disease too well here, but he did have small lesions in the liver. He got his doses of ipilimumab, and by week 12, this is what the liver looked like. So this was pretty scary, actually, because if this was chemotherapy, we definitely say the chemotherapy wasn't working. But this patient was actually feeling quite well, despite the scans, so we opted to just watch him. And again, 8 weeks later, no further therapy, this is what the liver looked like. Week 36, this is what it looked like. And this patient started treatment in 2006, and his liver looks the same now.
Quick question, Rich. Is there a correlation with the mutation of melanoma? Does it matter if it's BRAF?
So we're starting to look at that. Yeah, it probably is relevant, but we don't know yet. Yeah. So this is the graph that Ron just showed you. You know, because we think that the immune system and immune response is important, this led to the work that Ron did, thanks to the funding provided by this group. And I don't have to go through this again, but basically, as Ron showed you, although both KIT inhibition with a TKI, here Gleevec and here this drug dasatinib, as well as CTLA-4 therapy, you can get an initial shrinkage in tumor, but it just grows back later. But if you give both in combination, you get the shrinkage, and then you know, prolonged disease control. And so, really, the next question for us is will this do the same thing in people? And so we're in the process of developing a study to test this hypothesis. And what we want to do is a phase 1 study combining a KIT inhibitor. Right now our plan is for dasatinib with ipilimumab in patients with GIST. And this study would be the first study to ever combine ipilimumab with an inhibitor of KIT. So this would be a real novel therapy. Since it's the first time we'd be combining these two agents, so the primary endpoint of this study would be safety, to identify the safe and tolerable doses of drug. And our plan would be to give continual dasatinib. Ipilimumab is currently, you get it IV, and you get it once every 3 weeks for what we call these 4 induction doses, and then you get subsequent doses at 12-week intervals. And so that would be the treatment plan, and we'd have to start with relatively low doses and increase slowly until we've make sure that the treatment's safe. But not only do we want to see what doses are safe, we want to see does the drug work, and also in this trial it'd be very important for us to get tumor specimens as well as blood specimens, so we can actually test the hypotheses that led to this. You know, can we look at the CD8 to Treg ratio? Can we look at IDO levels within the tumor? Can we look at changes in the immune system that we think we should be doing? And so we would get biopsies from patients at baseline. Again, they're getting dasatinib for 2 weeks before they get their dose of ipilimumab. So then we can get another biopsy before the ipilimumab, so we can see what did the dasatinib do by itself, and then get another biopsy after the combination, and we begin blood throughout. So our secondary endpoints would be response rate, progression-free survival, and overall survival. And then Dr. DeMatteo, Dr. Wolchuk, and the Ludwig Institute would do our immunologic correlates. So I think that's all I want to kind of present right now. And certainly I think particularly this dasatinib ipilimumab trials is really it's an outgrowth of the depth of experience that we have here. You know, it's really amazing to have Christina right here and Ron right here, so we can just go to each other's office and run ideas and get studies like this up and running. So thank you very much. And any questions?
Why was the dasatinib chosen? It seemed to have done very poorly as a monotherapy.
Good question.
Choose a more successful.
I would love to. So
is the camera on?
yeah.
The Pfizer cynics here want to know if BMS funded it. Not that I was thinking it.
So the National Cancer Institute approved the trial already, okay? But that was, what, 3 months ago? The problem is that it's very difficult to get 2 different drug companies to provide their drug for a combination trial. So dasatinib and ipilimumab are made by the same company. So that's part of the rationale. I mean, dasatinib's never been tried. Well, the data on upfront dasatinib is not entirely clear. It's been tried as a second-line agent, but not upfront because imatinib is the upfront drug. So that's part of the answer.
How could we convince them that that's
Well, we're trying, but it's not easy.
Yeah, sometimes it's hard to coordinate between the NCI, the drug companies, and what we optimally want to do. You know, I mean, we need to design a trial that's feasible, that people are going to support, and that we think will answer the question. So I agree with obviously all of your concerns that, you know, if we could design our ideal trial, maybe this wouldn't be it, but I think this will still answer the questions that we need to answer.
So we're basically also need NCI to
Well, no, the NCI's already approved it.
Oh, okay.
It's that the drug companies have to give us the drug.
So it's only you that need to convince them that they will be working on the clinical trial and all that, and getting the drugs. Is that what it is?
Right. I need the drug companies to say yes, we will allow the NCI to give us the drug.
Is there anything that we as patients can do anything about it?
You know, patient advocacy has a tremendous amount of power.
Well, that's why I asked, could we petition it?